Our Director of Research, Dr. Colin Young, has been looking at the drug development pipeline. So how big is this space and what might we anticipate has been a huge driving question for all of our consortium members. Is it hundreds of products? Is it dozens of products? Is it only a handful? And how many patients, as you heard, are really going to qualify and be eligible for these? These are important questions so we know the size and the scope of the financing challenge that they might present. So, Dr. Young, would you like to share with us some of your team's findings?
Thank you, Mark. Good morning. I'm kind of the designated Grinch today. So I'll try and explain why I'm a Grinch, a little bit why I expect my Grinch-like qualities to modify as we go through time. And to do that, I'll tell you a little about the forecasting process, and then we'll look at some of the results of our forecasts. Our universe is a little smaller than the ARM universe. We look only at stuff that's likely to come to market in the US. And we don't look at the full range of regenerative. There are a lot of things that are outside our universe. We're looking at, and I'll say a little bit later, but I'll say it now, that this is a population. Is my voice disappearing? I started working with at the beginning of last year, and we've kept going with for a while. Things have changed extremely rapidly in this landscape over the last year. So we will find that if I talk a little bit about what the pipeline looks like going forwards, it'll look a little bit unlike the one that we've been working on. We'll talk about that later. Give you an upfront idea of some of our projections. We're looking by our horizon is 2030. So by 2030, we're looking at about 40 to 60 approvals. We're looking at a patient population that would be treated between 40 and 55,000. And we're looking at total implied drug cost of 20 to 25 billion. Does not include any of the ancillary costs. So if we were looking at a CAR T, we might believe the CAR T treatment costs 400,000 in terms of direct drug cost. We'd probably expect to see at least that amount more in terms of hospitalization, medical costs in general. So it's not a simple, straightforward matter. So let's talk a little bit about how we forecast. We use a Monte Carlo model. A Monte Carlo model means that we do very large numbers of simulations. We have a couple of key parameters that we allow to become stochastic, and we see what happens in each of those simulations. So we'll do many thousands of simulations, and the results come from the average results of those simulations. So we'll have some simulations which are, should we say, wildly optimistic, some which are pretty pessimistic. And we're looking at, when we give you a range of results, we're probably looking about the middle 80% of that distribution. We're looking at US only, so we're looking at forecasting drug approvals for the US market, and we're looking at patient populations within the US and implied costs within the US. Our drug pipeline was originally drawn from the Sightline Pharma Projects database, which is a very large database. It contains pretty much everything that goes from a glimmering of a hope of being able to produce a drug to actually drugs that are going through the clinical process. Having got that pipeline, we got trials data from both there, from another database that Sightline have called TrialTrove, and from clinicaltrials.gov. It's a very long-winded process. Each drug we model separately, and if a drug addresses more than one disease, and in some cases they may address 5, 10, 15 diseases, each of those disease lines will also be modeled separately. Our key model parameters are progress rates, that is how long it takes a trial to get through a particular phase. Success rates, that is what's the probability that it will succeed in that particular phase. And those are the parameters that we allow to become stochastic when we're doing the Monte Carlo simulations. We also look at eligible patient populations, and those tend to be modeled to reflect targeted genes or antigens. So we don't just look at, for example, 155,000 potential non-small cell lung cancer patients. We consider what the antigen is that is being targeted. We look at adoption inhibitors. That is, why aren't these drugs going to be instantly adopted? And we've heard the earlier panel talk a little bit about that. There's a fair amount of resistance at the patient level. There may be some resistance at the physician level. Payers are uncertain, and developers have capacity constraints. There are a lot of reasons why we might see slow adoption for some of these drugs. The last thing we have to have is some form of pricing assumption. As has been said earlier, pricing is not our thing. We don't figure out what prices should be, but we have to have some assumptions when we're going to try and figure out what total costs will be. So we'll talk about those. We've talked about durable and potentially curative therapies, and that's essentially what we're looking at. This is our universe. We're looking at therapies which work on an intracellular level and try and make changes that will be curative and durable. Mostly that means we're using a variety of gene delivery mechanisms. I'm not going to go through what an AAV is. In fact, it's kind of a cool picture. What we do know is that this is a complex process, but it's also an extremely well understood process at this point. If you Google AAV, you'll find half a dozen or more companies will pop up that are quite happy to take your little snippet of RNA and package it into a viral delivery vector. What's more, many of them will sell you the various bits and pieces you need to do it yourself. So you can do it in-house. You can do it in-house with the aid of equipment that's being sold off the shelf by companies such as Miltenyi. So it's very easy to set up an in-house line. That is why we're seeing so many trials appearing in very strange places these days. We haven't encompassed those very much in our current universe, but the next time around there'll be a lot more trials from a lot of strange places. AAVs are probably the most widely used mechanisms, and they're used primarily in vivo. So these are things that you can just inject into a patient one-off, and they will either counteract the effect of a genetic aberration or will convert targeted cells to perform a particular function. You get then, say, hemophilia, where you're trying to persuade liver cells to produce factor. Other delivery mechanisms, lentiviruses. Lentiviruses are basically a branch of retroviruses which include HIV. And typically when we're talking about lentivirus delivery, we're looking at a stripped-down HIV, which is replication deficient because we don't want it to turn into a raging virus. Most of these are used ex vivo. And so we talked about beta thal, and we're looking at modifying bone marrow. You probably use a lentivirus to do that modification to extracted bone marrow and then transplant the bone marrow back into the patient. The other wide use for lentiviruses is we're creating CAR-Ts and TCRs. Again, we talked about a little bit before about CAR-Ts, and I'll talk about more of them in a moment. These things are used in vivo as well to directly correct a genetic aberration. The difference between lentiviruses and adenoviruses is adenoviruses don't integrate into the genome. Lentiviruses do. Means in vivo you're taking a significant risk if you're using a lentivirus, because if you do something wrong, it's gone into the genome and it will replicate. So there have been relatively few lentivirus trials, but they do exist. The other mechanism that's becoming at least widely talked about is CRISPR-Cas9, which along with zinc finger and talons are the direct gene editing techniques. And they, again, as we go forward, will probably produce a lot of interest. Okay, so moving on, I'm going to talk about oncology first. There's basically two sets of things going here. We've got what we think of as gene therapies and oncological treatments. The oncological treatments are essentially CAR-Ts and TCRs. And if we think about leukapheresis, about how we generate a CAR-T, we take blood cells, white blood cells from a patient, we separate out the T cells, We activate them. We engineer them to put the specific chimeric antigen receptor that we need in them. So if we're talking about Kymriah or Yescarta, we're putting in a receptor that basically is designed to attach to CD19. We grow out the cells, we purify them, and then we stick them back into the patient. And the idea is that they then go and kill the cancer cells. In the case of CD19 and B cells, cancers in general, basically all B cells express CD19. So the issue there is that you're likely to kill the cancer cells, you're also likely to kill all the B cells. The nice thing is we can live without B cells. The issue we have in solid tumors is most of the time if you kill all the tissue, you can't live. So there are different sorts of problems. As has been said, these are second and third line treatments. We're looking at relapsed and refractory patients. So looking at a relatively smaller subset of the population. I'll talk about that in a moment. As we've said, Kymriah and Yescarta both focus on CD19. We're looking at acute lymphoblastic leukemia and diffuse large B-cell lymphoma. A lot of success. We're forecasting from the pipeline, we're looking at 20 to 25 additional products approved in this sort of area by 2030, and they'll be looking at acute and chronic leukemias, basically CD19s, B-cell lymphomas, which is the bulk of the bulk of the non-Hodgkin's lymphomas, and we've also said multiple myeloma, which address BCMA. We're looking at pricing because we're going to have to figure out a price point to see what the effect is going to be. Our price points that we can see are Kymriah is about $475,000, Yescarta about $375,000. For our purposes, we assumed a price point of about $400,000. The combined incidence of hematological cancers is around 150,000. It's not a gigantic number. About 70% of those are treated successfully by traditional treatment regimens, which means you've got about 30% of that population is going to turn out to be relapsed or refractory. And those are the potential candidates for CAR-T. The rough rule of thumb for relapsed and refractory is we look at the percentage of the population initially diagnosed that do not live for 5 years. If they live for more than 5 years, it's generally the case that they're in remission and doing okay. And we're forecasting that about 22,000 to 28,000 patients will be treated annually by 2030. Those are essentially the ones who are in the B-cell conditions. So it's B-cell, ALL, and B-cell lymphomas, and also the multiple myeloma. The annual direct cost of product from that, assuming that 400,000 price point, would be between $9 and $11 billion. To scale that, the total US prescription drug reimbursement cost last year was about $400 billion. So we're looking at a relatively small percentage. The thing to remember here, I guess, if we're sort of thinking about the future a little bit, if I think about what trials look like at the end of this year. This is not in our current population. This will be in the next round of forecasting. We know that at this point there are 220,000 active trials in hematological cancers. We know that 133 of those are targeting CD19. So we're going to look at an awful lot of products that think they're going to be Kymriah and Yescarta. There probably isn't going to be a whole lot of room in the market for them. Now, the good thing about this is an awful lot of those trials are taking place in China. Basically, Chinese hospitals, Chinese companies, research establishments. And most of this product is going to be aimed at China ultimately, because there simply isn't room here. The one thing we know is that 150,000 figure for total hematological cancers in the US isn't going to change. It doesn't matter if you've got 40 or 60 or 80 products. You're still looking at the same number of patients. So if I'm looking at a bottom line number, how much it's going to cost, this is where the big bulge is going to come in development of these treatments. and it's not going to make very much difference at all to total cost or to patients treated. Solid tumors to date have not been a success story, and I apologize for that, but that's just an observation. Yeah. We've had no phase 3 trials. We've had 18 products that have failed in phase 2. I can tell you that as of the end of 2018, end of last year, there were still no phase 3 trials. So there is nothing that has advanced particularly well. Our forecast is 1 or 2 products will have been approved by 2030. There are lots of reasons why that could be a bigger number, but right now, that's what we would look like. That's what we would look at based on parameters developed from what we've observed to date in terms of progress and success. Number of treatable patients would depend on the cancer addressed and the antigen that's being targeted. We've got an awful lot of trials looking at an awful lot of different antigens when we're looking at solid tumors. Each of those antigens there are being targeted by at least 20 trials currently active. All of them are being used for at least 5 different tumor types. So when we start looking at, okay, let's figure out one of these works. What's the patient population going to be? It's likely to be an awful lot smaller than the total potential population if you look at how many people get breast cancer, how many people you're actually going to be treated if you get an MUC1. You might be looking at 12,000 out of that 220,000 initial diagnosis. So overall, we're forecasting probably between 8,000 and 12,000 patients treated annually by 2030. Direct cost of product, $3 to $5 billion. Uncertainties come from the differences between the cancers. If we run a set of simulations where non-small cell lung cancer gets big hits, that's going to be a lot of patients. Anal cancer would be a lot different. We've got potential for multi-cancer success from antigen targeting. So we saw that there are a lot of cancers that express the same antigens. If you happen to get lucky with HER2, you may be able to treat, brain, breast, a couple of other cancers, but we don't know. Combination trials. People are starting to run trials which look at not only the CAR T, but also using something like pembrolizumab, which essentially suppresses the cancer's ability to hide. So if we can prevent the cancer from hiding, we have a much better chance for the CAR T to kill it. We don't know how those will succeed. There are a lot of being run. We have no data. Okay, enough about oncology. Gene therapies. We think about gene therapies in terms of the diseases they address, and the diseases they address we classify as either ultra-orphan, which are things with a prevalence of less than 10,000, orphan, the FDA definition is 200,000 or less, and everything else, the large therapy, large prevalence conditions. In terms of what we've got, we've seen a fair amount of success and not so much failure in the orphan diseases. We've seen little success and a lot more failure in the high prevalence conditions. That's not as marked as the case when we're looking at oncological, hematological versus solid tumors, but it is still marked. There's a lot less success in those bigger prevalence cancers. And mostly this is because as has been observed, most of the orphan diseases we're looking at monogene treatments. In terms of our forecasts, we're looking at 6 or 7 by 2030 in ophthalmology. We've already seen retinitis pigmentosa. We'll probably see macular degeneration. We'll see a couple of others. Hematology, the hemophilias, sickle cell anemia, beta thal, essentially all look pretty promising. Neurology, spinal muscular atrophy, certainly. ALS, maybe. Again, ALS is one of those conditions where you're actually looking at a very small proportion of the total population. You're just looking at people who have a problem with the SOD1 gene, which is a small fraction of the total ALS population. Metabolic, cystic fibrosis, Huntington's disease, and then all the others, which would include things like cardiomyopathy and hereditary angioedema, and a whole bunch of other conditions. It's a reasonable amount of success. We've had one success so far, which is Luxturna, which has been talked about. That addresses a small subset of retinitis pigmentosa and Leber's congenital amaurosis. That is mutations to the RPE65 gene. The total incidence for those conditions would be 1,000 for RP and 100 for Leber. In terms of the eligible incidence, about 6 of those 1,000 would have that particular genetic aberration for RP and about 16 for Leber's. That's a very small population. However, it's not a fatal disease, and there's a whole lot of people who have it. That's just per year, so there's a considerable prevalence. So the potentially eligible prevalence would be 450 for RP out of about 80,000, and about 1,100 for Leber's. So it's not huge numbers. That's relatively common among these conditions because I say, we're looking at particular monogene issues. For all gene therapies, again, the total eligible patients is going to be sensitive to the success of trials for the high prevalence indications. Our forecast is 12 to 15,000 patients annually by 2030. Again, if we had a hit in diabetes, if we had a hit in peripheral arterial disease, those numbers could go up quite easily. We don't have very much in the way of price points. We've seen Luxturna is priced at $850,000. That really doesn't tell us a whole lot. Our assumptions for pricing, for doing our estimates of total costs, are that ultra-orphans are going to be about 2 million, orphans about 1 million, and the high prevalence conditions about 500,000. Given those assumptions, then we're looking at an annual cost of product of between $8 and $10 billion in 2030. Okay, the end of this is what we can't forecast. Because of the nature of the process, we can't forecast unpredictable changes in success and progress rates. Those are the parameters that go into the forecast to begin with. We have no mechanism for forecasting changes. We can observe changes, so when we see changes, we can do a new forecast. But during the forecast, we cannot do that. Things that might drive those changes would be, we've got product groupings, so we know there's a lot of CRISPR-Cas9 stuff going on. If any of those were successful, that may well drive a lot more success in that particular area. Similarly, companies have platforms where they do a lot of similar trials for similar types of conditions. If any of those work out, you're likely to get an accelerated success. Patient availability is an issue, particularly if you're looking at really, really small incidence conditions. It's really quite hard to run a trial when you've got to gather enough patients to run a trial. It's a lot easier if you're running your trials in China. Much bigger population, much easier to get access. The other reason things might change is just through evolution in the industry. There's a lot of shared knowledge, and as we get better at doing things, we expect things might change. What happens if, suddenly we get, peripheral vascular disease, diabetes, all of those things with very high prevalences? What happens if they suddenly develop cures? It's an issue that we can't predict. We just assume there's a normal probability that that'll happen, but in real life, things happen. These can be mitigated, so something like Alzheimer's, which has an incidence of about 1.5 million. The only things that we're actually able to address right now would be early onset, or what's known as APOE44. If we got either of or any of those right, each of them is about 2% of the population. So you're looking at about 30,000 rather than 1.5 million. Relatively small numbers. What's not in our forecast, pipeline growth. I've talked about that as we've gone along, particularly in the oncology area. Same thing holds in gene therapy, except there's been very, very much slower growth in gene therapy. Over the last year, there's probably maybe 10, slightly more than 10 new programs, trials programs come in gene therapy. So it's a relatively slow rate of change compared to oncology. The other thing we haven't talked about is offsetting costs. We know that a lot of these therapies, a lot of these diseases have therapies right now which are expensive. And if we look at hemophilia, the average cost for prophylaxis for hemophilia A is about $350,000 a year. And when you're saying what's the cost of a hemophilia gene therapy, well, you're looking at maybe $1 million or $2 million, but you're also looking at not having to pay $350,000 a year for the rest of the patient's life. So if it's a successful treatment, the offsetting costs are really important. And those sorts of costs appear for a lot of therapies. What else have we got? Okay, just to go back to the beginning, the pipeline of gene therapies in development, we were looking at 425 drugs, 214 diseases. Our projection is 40 to 60 approvals by 2030. Resultant patient population treated of 40 to 55,000 per year, and a per year implied cost of 20 to 25 billion. Okay, and that I think is it. I don't think, am I out of time, Mark? You are. Yeah, I'm well out of time, so I get to do the finally thank you.